Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. My name is Muhammad Kamal Khan and I am the microbiologist. Today in this video lecture we are going to talk about the difference between gram-positive and gram-negative bacteria. In this video lecture we are going to talk about the difference between gram-positive and gram-negative bacteria, and these are the main content of the gram-positive and gram-negative bacteria. So we are going to talk about the difference between gram-positive and gram-negative bacteria by their definition, cell wall thickness, peptidoglycan layer, teichoic acid, porins, mesosomes, morphology. So we are going to talk about the differentiation between all these with gram-positive and gram-negative bacteria. Moreover, we are also going to talk about the difference between lipopolysaccharide, toxin produce, resistance to antibiotic, examples, gram staining, flagella structure, lipid content. So we are going to talk about also these main content as well in gram-positive and gram-negative bacteria. So let's talk. First of all, let's explain the differences. Let's explain the gram-positive and gram-negative bacteria by diagrammatically. Okay, so this is the first diagram of the which are actually explaining the gram-positive and gram-negative bacteria. Okay, now under the light microscope, what actually you are going to do when you stain the colony, let's suppose you have a colony onto the slide, on a clean slide, and when you stain it with a gram stain, inshallah in some other video lecture we will explain the gram staining technique. So gram staining technique actually nothing but it's actually the staining which we perform the staining on the slide. So what actually we are going to do, if we stain the slide with a gram stain, so after that if we want to see it under the light microscope, so under the light microscope the gram positive bacteria will be appear as a bluish purple in color, as you can see. So this is the gram positive bacteria, so it appear bluish color, okay, bluish purple in color. And if you want to difference between gram negative bacteria, so remember guys, when you stain it with a gram stain, so then the gram negative bacteria will be appear reddish pink in color, as you can see. This one is the gram negative bacteria. So this is the diagrammatically differences between gram positive and gram negative bacteria. Moreover, we can also distinguish the gram positive and gram negative bacteria by their structure. As you can see, this is the cell wall of the gram positive bacteria. So this is the cell wall of the gram positive bacteria and this is the cell wall of the gram negative bacteria. So remember guys, in case of the gram positive bacteria, the cell wall is thick and there's many layer of the cell wall, many layer of the peptidoglycan, as you can see. And moreover, it have only one membrane, as you can see. So this is the only one membrane in case of the gram positive bacteria. And it have also protein in their cell membrane. But if we compare it with the gram negative bacteria, so remember guys, in case of the gram negative bacteria, we have lipopolysaccharide, we have Porin channels, we have outer membrane, we have peptidoglycan layer as well. But in this case, the peptidoglycan layer is a single layer peptidoglycan layer, okay, or we can say it's a very single, or okay. So in this case, the thickness of the cell wall is not high as compared to gram positive. Then we have also an inner membrane and some protein are present in the membrane as well. So we have also some periplasmic space which are found between this cell wall and this cell membrane. We have some periplasmic space. So remember, yeah, this periplasmic space is not only available in the gram negative bacteria but also in the gram positive bacteria. But in gram negative bacteria we have more periplasmic space as compared to the gram positive bacteria. So this is actually the diagrammatically explanation of the gram positive and gram negative bacteria. Then let's explain our difference between the gram positive and gram negative bacteria by the introduction. First of all, what is gram positive bacteria? Gram positive bacteria are that bacteria which have thick cell wall and under the night microscope it appear bluish purple in color when we stain it with a gram stain. Okay, and what is the gram negative bacteria? Gram negative bacteria are that bacteria which have thin cell wall and when we stain it with a gram stain, then under the light microscope it appear reddish pink in color. Okay, so let me show the diagram. As you can see, this now as you can see, this one is the gram positive bacteria which is bluish purple in color under the light microscope when we stain it with gram stain, and this is the gram negative bacteria which is appear reddish pink in color under the light microscope when we stain it with a gram stain. Okay, so this is the difference between gram positive and gram negative bacteria. Then let's difference between the gram positive gram negative bacteria by their cell wall. So remember guys, gram positive bacteria have smooth and double layer cell wall, or it have a double layered cell wall, while in case of the gram negative bacteria, the cell wall is a single and wave cell wall, okay, not a smooth cell wall, we can say like that. So as you can see, this one is what this is actually the cell wall of the gram positive bacteria. As you can see, is a very thick and moreover is a smooth cell wall. And if we see, this is the cell wall of the gram negative bacteria. So as you can see, the cell wall of the gram negative bacteria is a very thin and not a smooth like, okay. So this is the difference between the cell wall of the gram positive and gram negative bacteria. Then we have the cell wall thickness between gram positive and gram negative bacteria. So remember guys, the thickness of the gram positive bacteria cell wall is 20 to 80 nanometer, and the thickness of the gram negative bacteria is 8 to 10 nanometer thickness. It means that gram positive cell wall thickness is more higher as compared to the gram negative bacteria. Then we have the peptidoglycan layer. So remember guys, in case of the gram positive bacteria, there are many layers of the peptidoglycan. That's why the cell wall is thick. But in case of the gram negative bacteria, the peptidoglycan layer is a single peptidoglycan layer. That's why the cell wall is thin in case of the gram negative bacteria. Then there is a question arise in our mind that what are actually the function of this peptidoglycan layer? Remember guys, in gram positive and in gram negative bacteria, the most important function of this peptidoglycan layer that it prevents the bacteria from environmental stress. Moreover, it also prevents the morphology of the bacteria as well. Okay, so this is the function of peptidoglycan layer. Then we have teichoic acid and outer membrane. Okay, so remember guys, in case of the gram positive bacteria, teichoic acid is present but the outer membrane is not present, as we explained in the in the diagram. As we explained in the previous diagram, and if you watched that, what we actually explained that in case of the gram positive bacteria, there is no outer membrane due to the thick of the cell wall. But in case of the gram negative bacteria, there is a cell membrane, outer cell membrane, which we call that, and it is all about the thickness of the cell wall. Okay, so in this case, the teichoic acid is present but the outer membrane is absent. Okay, and in case of the gram negative bacteria, teichoic acid is absent but outer membrane is present. Okay, now there is a question arise in our mind that what are actually the function of the teichoic acid and outer membrane. Now what are the function of this teichoic acid and outer membrane? Okay, first of all, let me show the diagram. This is the diagram. Okay, this is the diagrammatically explanation of what? This is the diagrammatically explanation of teichoic acid. So remember guys, this one, as you can see, this is actually the teichoic acid, and there is also some other teichoic acid which we call it lipoteichoic acid. So whenever the teichoic acid, as you can see, this teichoic acid are actually connected with some other things. And remember guys, there are also some protein which we call it wall associated protein, as you can see. Moreover, it is a cytoplasmic membrane, as you can see. But remember guys, this is actually the gram positive bacteria cell wall structure. Okay, now what is actually the function of teichoic acid and outer membrane? The function of the teichoic acid, it provides the flexibility to the cell wall. Now why? Because inside the cell wall, not only the teichoic acid is present, there are also some electrolyte, like we can say calcium and potassium are also present. So due to the attraction of calcium and potassium, which is actually nearly present to this teichoic acid, so what actually going on due to the attraction of these two calcium and potassium, so what happened, the cell wall flexibility is maintaining. So this is all about the teichoic acid function. Then we have outer membrane function. So remember guys, the outer membrane function is to protect the cell from outer environment. This is actually the difference between teichoic acid and outer membrane. Then we have porins and lipopolysaccharides. So remember guys, this is, as you can see, this is actually the diagram. And remember guys, this is the porin, as you can see, this red color. Now this red color, as you can see, this is actually the porin channels. Okay, so remember guys, the porin channel is present in case of the gram negative bacteria. Okay, and porin is always available at the outer membrane surface. So this is and this is actually the lipopolysaccharide, as you can see. This is the lipopolysaccharide. So what are actually the porin function and lipopolysaccharide? Now what are the function of this lipopolysaccharide? Remember guys, the main function of this lipopolysaccharide is to maintain the permeability of the cell membrane. Okay, so it actually helps in the permeability of the cell membrane. So all those content which the cell needed, so the cell will receive all these content through the cell membrane by this lipopolysaccharide. But if the content which the cell do not need, so these things will be remain outside. So this is all about the lipopolysaccharide because it maintains the cell permeability. Moreover, we have the Porin channel. Remember guys, in case of the gram-negative bacteria, the function of the porin is to protect the cell from the antibiotic. So what actually going on, if the antibiotic, let's suppose, get inside the cell, so what happen, it will again release out from the cell through this Porin channel. Okay, so we can say that gram-negative bacteria is actually more resistant as compared to gram-positive bacteria from antibiotic. So in case of the gram-positive bacteria, porins and lipopolysaccharide are absent. That's why we can say that gram-positive bacteria is actually more susceptible to antibiotic. But in case of the gram-negative bacteria, this is actually gram-negative bacteria. Okay, mistakenly write it. This is actually gram-negative bacteria, not gram-positive. In case of the gram-negative bacteria, porins are present, but porins are present at outer membrane, and lipopolysaccharide are also present. Okay, so this is all about the porin and lipopolysaccharide in gram-positive, gram-negative bacteria. Then we have mesosomes and lipid content. Remember guys, in case of the gram-positive bacteria, mesosomes are more prominent, means are more efficient and are more effective and active. In case of the gram-negative bacteria, mesosomes are less prominent. Okay, then we have the lipid content. In case of the gram-positive bacteria, lipid content are very low, and in case of gram-negative bacteria, lipid content are 20 to 30%. So this is actually the diagram which is actually showing us the mesosomes. So as you can see, this is the mesosomes. Okay, so it is more rare in case of the gram-positive bacteria, but less rare in case of the gram-negative bacteria. Now what are the function of this mesosomes? So as it is more prominent in case of the gram-positive bacteria, because the mesosome function is to form the cell wall. It actually help in the cell wall formation. As it have thick cell wall, that's why it help in the cell formation. In case of the gram-negative bacteria, the mesosome concentration are very less or very less prominent. That's why we can say because in this case the peptidoglycan layer is very thin, or we can say the cell wall is very thin. That's why in this case the mesosome are less prominent. Now what are the function of this lipid content which are less in case of this? Lipid content is actually mainly responsible in the cell wall cell membrane formation. Okay, so in case of the gram-positive bacteria, as there is only one means there is only inner cell membrane, so that's why lipid content are less available. But in case of the gram-negative bacteria, there is inner membrane as well as outer membrane, that's why the lipid content is 20 to 30%. Okay, then we have morphologically explanation between gram-positive and gram-negative bacteria. So remember guys, this is the gram-negative bacteria, morphology of the gram-negative bacteria. In gram-negative bacteria, we have, gram-negative bacteria are also in a cocci shape. As you can see, cocci means a round shape. So bacteria, the gram-negative bacteria are also in a diplococci shape, also in a streptococci shape. Okay, as you can see. So if you found in under the microscope, if you found that gram-negative bacteria purple in color, and moreover you also found a round shape or cocci in shape, so remember, and the cocci are actually diplococci, means two cocci are connected to each other, so we call that Neisseria meningitis. Okay, this bacteria is called as Neisseria meningitis, but still it is not confirmed because you have to perform biochemical test to confirm it 100%. Okay, inshallah in some other video lecture we will explain the biochemical test in detail. Then we have Streptomoly Formas. Okay, in this case as you can see, we have a rod shaped bacteria. So this is actually the gram-negative bacteria are also in a rod shape, but if there is a lot of rod shape shapes are connected to each other, so then we will call that Streptomoly Forms. Okay, so this is the another morphology of the gram-negative bacteria. Then we have rod shape of the gram-negative bacteria, as you can see, and we call that E. coli. So under the light microscope, if you found pink, reddish pink color, and the shape is a rod, so remember guys, that will be the E. coli. But still you are not 100% sure, still you perform some different biochemical test. Now under the light microscope, if you found this helical like structure, so we call that Treponema pallidum, and it will be appear in a pinkish red in color because it is gram-negative bacteria. And if you found this like that shape, as you can see, so remember guys, we call it spiralum volatum because it look like a spiral in structure. And this one structure is called a helical structure, and this bacteria, if you found it under the light microscope and if it is pinkish in color, so remember guys, it is Helicobacter pylori. Then we have some other diplococci, as you can see, so we call that Neisseria gonorrhea. Then we have vibrio like shape of the gram-negative bacteria, which we call a vibrio cholera. Okay, so this is actually the morphology of the gram-negative bacteria. It doesn't mean that under the light microscope, if you found all these bacteria structure, it doesn't mean that all these bacteria will be the same bacteria as I write the name here. Now still you have to perform different biochemical tests. Through this biochemical test, you will 100% sure that what type of bacteria is it. Okay, then this is the gram positive bacteria because under the light microscope it appear in a bluish purple in color. Okay, so remember guys, if you found round cocci or cocci, but the cocci are connected to each other, so this is called a Staphylococcus aureus. So mainly Staphylococcus aureus are cocci in shape or round shape. Moreover, we have also some round shape of the bacteria, we call it Streptococcus pneumonia, as you can see. Then we have another structure of the gram-positive bacteria, another morphology, which have both rod shape and cocci shape connected to each other. We call that Chronium bacterium diphtheria. Okay, then we have also the same in case of Clostridium botulinum. They have also cocci plus rod shape, so connected to each other. Then we call that Clostridium botulinum. Then we have also cocci shape of the bacteria, we call that Micrococcus luteus. And remember guys, Bacillus anthracis, in case of Bacillus anthracis, remember guys, if you found purple color under the light microscope and all the rod shape you find it and it make a chain, as you can see, so we call that Bacillus anthracis. But still you perform biochemical test. Then we have Streptococcus pneumonia, as you can see, which are actually the cocci and connected to each other. And we have Pseudomonas Aeruginosa. So Pseudomonas aeruginosa is actually gram-negative bacteria, but mistakenly they draw here the Pseudomonas at that diagram. This is not actually the right. Okay, it is actually gram-negative bacteria. It will be available at that way. Okay, anyway, so this is the morphology of the gram-positive and gram-negative bacteria. And then we have the gram staining of the gram-positive and gram-negative bacteria. Remember guys, gram-positive bacteria, actually how the gram-positive bacteria appear. The gram-positive bacteria appear bluish purple in color under the light microscope. Now why? What's the reason behind that? Because the cell wall is thick and it retain the crystal violet. Crystal violet is actually the part of the gram stain. Inshallah we will explain it. And when it retain the crystal violet, so that's why it appear bluish purple in color. Okay, moreover, acetone and alcohol are washed out in this case. Now in case of the gram-negative bacteria, as the cell wall is very thin, so in this case crystal violet are not retained and the acetone and alcohol are washed out, and then under the light microscope it appear reddish pink in color. So again, this is the diagram, as you can see. So this one is actually the gram-positive and this one is the gram-negative bacteria. Then we have the flagella structure of the gram-positive and gram-negative bacteria. So remember guys, in case of the gram-positive bacteria, they are, first of all let me show the diagram. Now this is the diagrammatically explanation of the gram-positive and gram-negative flagella. So remember guys, this one is actually filaments, okay, and this portion is actually called as hook. This filament is actually rotating and this hook actually provides support to this filament. Moreover, we have basal body, as you can see, this is the basal body, and this basal body contain rings, And these rings, whenever the rings are rotating, so then this filament are also rotating. So this is actually the whole structure of the flagella. So remember guys, in case of the gram-negative bacteria, we have 4 rings in the basal body, as you can see. But in case of the gram-positive bacteria, we have 2 rings. So what does it mean? It means that gram-negative bacteria are fastly moving as compared to gram-positive bacteria. Then we have the example of the gram-positive bacteria. So we have Staphylococcus, Streptococcus, and some other bacteria as well. Then we have, in the case of gram-negative bacteria, we have E. coli, Escherichia, we have Salmonella, we have Pseudomonas Aeruginosa. Okay, all these are actually the example of the gram-negative bacteria. Then we have resistance to antibiotic and toxin produce. So remember guys, gram-positive bacteria are more susceptible to antibiotics. Now why? Because we talked that in gram-positive bacteria there is no porin and there is no lipopolysaccharide. That's why it is more susceptible to antibiotics, because porin is actually remove of the antibiotic and lipopolysaccharide is actually allow all those content which are needed for the cell, as antibiotic are not needed, so that's why it is remain outside. So what actually happening? So gram-positive bacteria are sensitive or susceptible to antibiotic, but gram-negative bacteria are resistant to antibiotic. Why? Because in this case they have porin, they have lipopolysaccharide, that's why it is resistant. Moreover, in case of the gram-negative bacteria, it produces endotoxin as well as exotoxin. Inside the cell they are producing the toxin, and outside the cell they are producing the toxin. And in case of gram-positive bacteria, it produces the toxin outside the cell. So still both are dangerous. So that's all about the differences between gram-positive and gram-negative bacteria. I hope you like this video. If you like this video, so make sure to hit the like button, share this video, and also subscribe the channel, and also press the bell icon. If you have any kind of the question in your mind, you can write your question into a comment. Inshallah, I will try my best to give you answer as soon as possible. So thank you so much for watching that video.